Hello everyone, welcome to Ant Biology webinar series. As you all know, this has been started by Translational Outcome Research Group of Department of Zoology, University of Calcutta, and we generally organize these fortnightly lectures on Saturdays and Sundays. I am Shohili Biswas, today's moderator. We are very much delighted to have Professor Gaurisankar as our today's speaker. We wholeheartedly welcome you, sir, on our platform.
Thank
And
you.
to our audiences, you can post your questions on the comment section below and we'll moderate them at the end of the talk. And continue till the end to know more about the talk the speaker is going to say. Hope you will enjoy the talk and get enriched with the talk. And now, I am without further delay, I am requesting our mentor and convener, Professor Ena Rai Banerjee, to introduce today's speaker, Professor Gaurisankar Sa, to our audiences. Over to you, ma'am.
So nice to see you, Gorida. A
Thank
very
you.
warm welcome to all our speakers. And like it happens every time, something that we do not have any control on, that is the internet connection. So just when Shohili had started the program, I found that my internet was acting up. So I'm a little, under trepidation with that, but nevertheless I always consider it as a good omen that something like that happens at the beginning of the show. And
Yeah.
today our speaker being Professor Gaurisankar Sa, it promises to be a riveting talk and a bit of a roller coaster ride also. So with that, I would like to introduce, it is my singular privilege and honor to introduce our today's speaker, and I will read from a very short introduction, which does not do justice to our speaker, but I think most of our audiences today and some of the new people who may have joined our program tonight would surely know him by name and his work. So here goes. Professor Gaurisankar Sa, Fellow of National Academy of Science, India, Fellow of Nanyang Academy of Science, Singapore, Fellow of West Bengal Academy of Science and Technology, received his doctoral degrees in biochemistry from the University of Calcutta in 1989. Then he joined Virginia Tech, USA as visiting faculty. Thereafter, he moved to Cleveland Clinic, USA to work on cell signaling. In 1994, he joined Bose Institute, India, where he is now working as a professor and chairman of molecular medicine. He is also a visiting professor of the Cleveland Clinic, USA. Professor Sa is the editor-in-chief of International Journal of Immunology, sectional editor of scientific reports, Future Oncology, PNAS India, editor of Journal of Cancer Research and Molecular Medicine, Austin Journal of Clinical Immunology, and many others. He is also the editorial board member of scientific journals like International Journal of Cancer Research, Journal of Pharmacology and Toxicology, Advances in Modern Oncology Research, etc. Research work of Professor Sa is focused in the area of cancer biology and tumor immunology and understanding of its molecular mechanisms. His work basically aimed at development of safe and non-toxic anti-cancer drug and development of adjuvant immunotherapy. His recent work also aims at understanding the mechanisms of immune evasion of cancer so that immunotherapy can be reinforced into cancer patients. His findings are highly recognized by various national and international scientific communities, as is evident from his more than 150 publications in high-impact journals, receiving various national, international awards and granting of a number of patents. So this by no means does justice to you, Gorida, who is a scientist par excellence and a leader in many a field of which he has been a vanguard. With that brief introduction, I warmly welcome you on our platform tonight. It is our privilege to host you, and if you would like to start your talk, We are all agog to listen to the latest findings from your lab.
Thank you, Anna, for such a nice introduction. And thank you, Sally, for moderating the program and communicating with me regarding this webinar. Without wasting time, I think that both of us has some pre-commitment to do some other things. I would like to start. So, shall I start?
Yes, sir.
Okay. So, my slides are visible?
Yes, sir. It is visible.
Okay. Thank you very much. Uh,
Thank you.
so, the title of my talk seems to be Cancer Immunotherapy. But basically, I'm a little bit of offbeat scientist. Most of the scientists that they think the cat should be kept in the box because that's the tradition. So most of the scientists they do the traditional research. But I leave the cat out of the back box because if you keep the cat out of the box, you will be able to understand the nature of the cat, whether it's a foe or a friend. Similarly, in my research area, I would like to do a out of box research. So the title of my talk is now Cancer Immunotherapy and Beyond. Before starting the talk, I would like to recognize the people who did real the most important work of my talk. My all-time collaborator, Professor Tanya Das, my clinical collaborator, Dr. Deeptendra Sarkar, and my overseas collaborator, Professor Jim Finke. And the work today I'm going to discuss is basically done by Abir, Sakib, Sayantan, Subhadeep, and Subhangi. The work done by other people in my group, I'm not going to discuss. As you know, this is the scenario of cancer. As you know, till date in 2010, nearly 1.4 million new cancer cases are happening every year, and around zero point 0.6 million cancer deaths are occurring in every year. So, if you look back from the development of various drugs, and as a result, the mortality rate in different disease reduced drastically. If you look at the heart disease, the if you consider 1950 versus 2010, the mortality rate in heart related death decreased dramatically. Similar scenario we can see with the cerebrovascular disease or infectious disease. But if you look at the mortality rate in cancer, rate in cancer, you will see even in 60 years, the mortality rate didn't change because of the discovery of different treatment procedure, but the death rate of cancer didn't change much until the new discovery in the last decades which is called the immunotherapy of cancer. Because of the discovery of various modes of immunotherapy, you see the mortality rate decreased nearly 20% in cancer. So what is immunotherapy? This is for the most of the students. So if you look at this graph, this is the tumor. If someone developed tumor, this is the tumor size with the time, how the tumor developed. And when the tumor becomes nearly 8 to 10 millimeter in diameter, we can recognize that there is a development of some lump in our body. We go to the doctor or clinicians. The doctor, they apply some chemotherapy. We can hope that the tumor volume will go down and you will get cured. You will be cured from the cancer. But the problem is that the after sometimes when the after the chemotherapy, when the patient goes to the holiday, the tumor again come back. This is called the tumor relapse. And similarly, when the tumor relapse, the patient goes to the doctor and doctor applies the second degree of chemotherapy, which is more harsh than the first degree of chemotherapy, with an hope that the tumor will go down. But within short period, the tumor again relapse, and some of the patient they die. And those patients, develop the second relapse, they go to the doctor, and doctor apply a very harsh chemotherapy, and the tumors little bit relapse, a little bit reduced, but again come back, and ultimately the patient die. So with the hope of the treatment procedure of immunotherapy, the doctor, the clinician apply the immunotherapy either at this juncture during the first chemotherapy, or when the tumor mass goes down dramatically and there is a visible reduction of the tumor. At that time, they apply the immunotherapy with an hope that the tumor volume will goes down and patient will survive. That's why the different immune therapy were discovered, and one of the noted immune therapy is the anti-CTLA4 therapy and anti-PD-1 therapy. For that, the Hozo and James Allison both got Nobel Prize in 2020. So, what is this immunotherapy? This is nothing but the development of some specific antibody towards these two molecules, PD1 and CTLA4. If you can mount these two molecules, then our immune system will be survived, so that our immune system can attack the tumor. By that way, the immunotherapy is applied. So, but the reality, what is the reality? If you look at, this is the metagenomic data that we received from all over the world, and we analyze this metagenomic data. So if you look at, so if someone developed cancer, and if that person was not given the any therapy, you will find the patient will die within 1 year to 2 years. So the 100% mortality in that cases. But if you apply chemotherapy, you see there is a change in the mortality rate, and the sum of the patient, nearly 5 to 10% patient, they survive till the 5 year. So 5 year mortality rate is survival rate is around 5% in case of cancer patient if you apply the chemotherapy. Nowadays, the genomically targeted chemotherapy, that means the patient are diagnosed with the mutation of different types of mutation which is being occurred in the cancer patient. And then by analyzing this mutation pattern, the clinician, they prescribe different drugs, and accordingly, the patients were treated. So if you see, if you go for the genomically targeted chemotherapy, the survival probability of the patient is better. And even the 5 year survival is made more than the 10%, more than 10%. But if you apply immunotherapy, you see the survival probability for the 5 years increase very dramatically. It increase up to nearly 20 to 25%. That's why the people are more people are looking for more immunotherapy for the treatment of cancer, and are recommended, cancer patients are recommended for the treatment of the immunotherapy. But you have to remember, the cost of the immunotherapy is enormous. The cost of immunotherapy is a perfect immunotherapy is around 2 crore rupees per patient. But if you look at the survival probability of this patient, if any patient is going for the immunotherapy, you see his survival probability is 1/5. So after using so much of, after spending so much of money, your survival probability is only 20%. So people are trying to understand why these patients are responding to the immunotherapy, how this can be improved. So as you know, I do my research in a quite different way, out of box research. So I am not interested to do the research on why this patient are, they are, why this immunotherapy is successful to this patient. I was interested to understand why this patient, they are not not responding to this immunotherapy? What is the reason why the immunotherapy is not working? So my entire talk will be related to the patient, why they are not responding to the immunotherapy. Can we change this pattern? Can you improve this pattern, this sensitive to immunotherapy from 20% to 70 to or 80%? That's my research. So for that, I would like to take you a overview of the tumor. So this is one of my favorite slides. This is nothing. This is not a galaxy slide. This is a tumor microenvironment. If you dissect the tumor and stain with different molecules, you will find that this is the nature of the tumor. And very interestingly, if you look at these blue dots, they are around 30% of the total tumor volume. And these blue dots are nothing but the cancer cell. So in a tumor mass, only 30% of the total mass is the cancer cell. What are the rest of the cells? What are the rest of the mass? Basically, rest of the cells are nothing but most of them are immune cell, some of them are endothelial cell, fibroblast cell, and some stromal cells. So, what is why these cells are there? Why these immune cells are there in the tumor microenvironment? What is their role? So, lots of work has been done in this field. And Bob Schreiber described the reason for the immune cell in the tumor microenvironment very nicely. What is the role of this tumor immune cell? So this is a tumor normal tissue. After we are exposing all the time with different types of carcinogens, viral infection, or some chronic inflammation, that are sufficient to convert this normal tissue into a transformed cells, and these are the initiation of the tumor. So when there is a transformation, our body has a very nice immune system, like different types of T cells, NK cells, and other cells, CD4 and CD8 T cells, macrophage, dendritic cells. When this immune system find that there is a abnormality in the cell, or there is a transformation of the cells, our immune system infiltrate in that sites, and they attack this tumor and try to to eliminate these cells. And those people who are lucky, this immune system can successfully eliminate these cells, so we don't develop cancer. So all the times when we are exposing to different types of cancer, different types of carcinogens, we don't develop cancer. That because we have a very good immune system in our body, and that protect us. Sometimes these mutations in these transform cells are very severe. So these cells became more smart, and what they do, they try to hide the antigen of this transform cells. So and they mount them so that our immune system cannot recognize these cells as a transform cells. And that way, these cells remain in our body for longer time, sometimes for 20 years. And whenever there is a problem in our immune system, that's maybe because of the aging, or that may be because of the overuse of antibiotic or anti immunosuppressive drug. Whenever these cells, these hidden cells, they see that there is some immune suppression in our body, these cells then start proliferating very fast and overpower our rest of the immune system which are existing in our body, and they modulate them. And this transformed immune system, they help the tumor to grow. So by that, in that case, then so when you will find that someone developed tumor, so it has to pass through all these processes. So our immune system plays very important role in the development or eradication of the tumor. So my research is basically concentrated on the third part, which is the escape part, how this immune system is playing role in the development or eradication of the tumor. So we have we try to understand how this how tumor modulate our immune system. So tumor modulate our immune system in different way. One of the way is this tumor secretes lots of factors that kills our immune cell, like particularly the cell mediated immune system like T cell. Effector T cell, they kill the effector T cell so that the effector T cell cannot activate the other immune system to eradicate or to attack the tumor. It blocks the cytotoxicity, T cell mediated cytotoxicity. Very interestingly, it blocks the it blocks the memory T cell, so that the memory, so our immune system cannot recognize that this is a foreign body or this is a transform cell. So they erase our memory to understand that these transform cells are cancer cells. They block the T cell proliferation so that the T cell number cannot increase, and they cannot attack the tumor. So various way they suppress our immune system. And very interestingly, there is one phenomenon which is called cytokine storm. They change this tumor change from the type 1 to which is a good cytokines to type 2 cytokine bias. So the Th1 type 1 cytokine producing cells number goes down, and type 2 cytokine producing cells number goes up. But by that way, they help the tumor to grow. And another way they help the tumor to grow is by augmenting the T regulatory cell number. So I will come to the details of this part in my talk. So our hypothesis, why the reason for the failure of immunotherapy is that if you have a tumor, we apply chemotherapeutic drug or apply genomically targeted chemotherapy, and we kill all the cancer cells. But and for that purpose we published. But very interestingly, a very small proportion of the cancer cell, they are drug surviving cells. They cannot be targeted by the chemotherapy or genomically targeted chemotherapy. And these cells are very multi-drug resistant cells. These cells are also called the cancer stem cells. So these cells, when we do the chemotherapy, these cells again start proliferating, and that's the reason for the immunotherapy, tumor relapse. So with the hope that our immune system can recognize these cells, and they can immune system can recognize that cell. That's why we start the immunotherapy by different way. One is the by adoptive transfer of chimeric T cell, or by applying the anti-PD-L1 or CTLA4 therapy, we can boost our immune system. And this boosted immune system can target our drug surviving cells, so that they can target this drug surviving or cancer stem cells. So there is a blockage of the tumor relapse. This is happening in nearly 20 to 25% of the lucky patient, which can respond to the immunotherapy. But what is going on into the other 70 to 80% patient? Very interestingly, in this patient, this tumor cell during the chemotherapy, these cells, they secretes lots of they try to resist the chemotherapy, and they secretes number of factors. And these factors are very important because these factors change the status of the immune system. Because we have a very active immune system, which is called the immunogenic system. They change this immunogenic system into tolerogenic system. Tolerogenic means they are transformed into type 2 immunogenic system, and this type 2 immunogenic system, they cannot attack this tumor cell. They cannot attack. Only thing on the other hand, they help the tumor cell to grow very rigorously and very fast, and they are more aggressive. So that's why the patient who are not responding to the immunotherapy, if you apply them to immunotherapy, the cancer become very aggressive and the patient die within couple of months if the immunotherapy fails. So that's what is happening. So our intention is, can we rejuvenate this tolerogenic immune system into immunogenic system? If we can re-educate this immune system from a type 2 to, if you can convert into type 1 cell again, then this immunotherapy will be more active. Then this immunotherapy will be able to protect this immunogenic cell after rejuvenation, and this immunogenic cell will be able to target our drug surviving cancer cell. In that case, we will be able to get the tumor, we will be able to kill the tumor. So, maybe in this 70 to 80% patient, this is happening. So, how can we rejuvenate this immune system? So before knowing this, how to rejuvenate, first we have to understand how these immunogenic cells are becoming the tolerogenic. So for that purpose, we did lots of research work, and the outcome of this research work is that we identify in new types of T cells that are developing during the tumor. So in a breast cancer tumor microenvironment, we identified a new lineage of IL-10 producing T cell, which is called T regulatory cell in tumor microenvironment during the development of tumor, and these cells increase during the chemotherapy. So we use by high dimensional flow cytometry and a tissue analysis. By that point, we have identified a new types of T cell which are pro-tumor. They help the tumor to grow. And you see, this is a pattern of this T cell in normal patient, and if you look at this pattern in cancer patients, you will find a specific types of cell which are predominant in tumor, which are tumor specific, and these cells are produce high level of CTLA4, another immunosuppressive marker, which are produced by this T cell. So what are these T regulatory cells? These T regulatory cells are specialized subpopulation of T cell that suppress the immune activation, thereby maintain in our body the immune homeostasis and prevent the pathological self-reactivity. Generally, in our body, we have a special types of T cell, which is called T regulatory cell, and the percentage of these T cells are very low, nearly 0.2 to 2% in our with respect to total T cell population, and these are the natural T regulatory cells. But this T regulatory cell has a dual role. It has some beneficial role in normal condition. These Tregs are necessary for the prevention of your autoimmunity. The Treg cells mediate immune tolerance. That's why our immune system doesn't attack our body cells. Those people who have the autoimmune disease, they are devoid of these T regulatory cells that are present in our body. But in cancer patient, the number of T regulatory cell increased dramatically from 0.2% to nearly 30%. The number increased dramatically, and this upsurge in tumor microenvironment is highly correlated with the poor prognosis of the cancer patient. So if this number goes up dramatically, the patient doesn't respond to the chemotherapy and immunotherapy. So these T regulatory cells are immunosuppressive. They suppress our immune system by inducing the T cell death. They cause the T cell energy, which are the responsible for the immune suppression. They block the DC maturation, dendritic cell maturation. They decrease the co-stimulatory molecules so that our immune system cannot be activated and they cannot attack the tumor. So they modulate our immune system into a tolerogenic immune system. Very interestingly, not only they moderate to they suppress our immune system, they help the tumor to grow by changing the macrophage pattern from the M1 type to M2 macrophage. This M2 macrophage is pro-tumorous. They secrete lots of VEGF-like protein, which are responsible for the neoangiogenesis, which is required for the tumor growth. They produce lots of rankle. This rankle is responsible for the metastasis of the tumor, and they help the development of myeloid-derived suppressor cells, which are more high immunosuppressive. So by that way, this T regulatory cell suppress our immune system. And if you look at when the number of this T regulatory cell in patient is very low, our immune system can correct they can kill the transform cells. When these T regulatory cells are fluctuating in our body, that means sometimes increase and go down. At that time, our immune system and tumor they coexist in our body, and the tumor cannot relapse. But when this T regulatory cell increase dramatically, at that time there is an immune suppression and our tumor cell grow very fast, and that's the outcome of the cancer. So if you look at this, this is a high-grade tumor and this is a low-grade tumor. This patient is responsive to both the immunotherapy and chemotherapy, and this patient doesn't respond to the immunotherapy and chemotherapy. You see, in high-grade tumor, the number of this T regulatory cell in tumor microenvironment is huge, nearly 20% of the total tumor. But the patient which are responsive, this number is very low. And if you look at the overall survival of the patient, if you have this low T regulatory cell, then nearly the 20% of the patient they die. Nearly 80% of the patient they survive beyond 5 years. And if you have a high T regulatory cell, nearly 80% of the patient they die. So only 20% patient they survive. And even in the distance metastasis-free survival, only 5% of the patient they do survive beyond 5 years. Beyond 5 years, if you have the high T regulatory cell in your tumor microenvironment. So the now new challenge in cancer therapy, particularly in cancer immunotherapy, is to suppress the T regulatory cell. How can we suppress these tumor-specific T regulatory cells? Basically, the T cells are developed in thymus and they go to the peripheral circulation as effector T cells. And these effector T cells, in presence of tumor, the tumor produce lots of IL-2 and TGF-beta. They convert this effector T cell into a T regulatory cells. And this T regulatory cell express lots of chemokine receptor like CCR4 that help the migration of the T cell, this T regulatory cell into the tumor site and causes lots of inflammation, angiogenesis, metastasis, and anti-tumor response. So by that way, these T regulatory cells modulate our body's immune system and help the tumor to grow. So how can we stop the development of this T regulatory cell? There are a number of factors or number of drugs. If you combine all these drugs, you can stop the development of the T regulatory cell, so that our immune system you can help the immune system to come back to the normal condition. But in that case, the problem will be that because of the uses of a number of battery of inhibitor and antibody, the patient will die because of the toxicity. So we cannot use the combination of this drug to suppress this T regulatory cell to make the immunotherapy more responsive. So, we were trying to develop different types of inhibitor without any success. Then we tried to sequence, analyze these cells, this T regulatory cell by single cell sequencing, and we got to know some total RNA pattern, transcriptomes, and non-coding RNA pattern. in these cells. And first, we try to understand what are the types of transcriptomes that are expressed in this specific tumor-specific T regulatory cell. Very interestingly, we found that in T cell, if you look at the RNA pattern in T cell and T regulatory cells, the pattern is completely different in both the case. So we analyze this from different angles. One of the very interesting angles we found that most of the T cell, normal T cell, which are effector T cell, they utilize glucose as their major source of energy. In these T cells, the glucose metabolic enzymes are expressed, highly expressed. That's why they use the glucose as their main energy source, and they don't use the fatty acid as their energy source, because the fatty acid metabolizing enzymes are down-regulated in T cells. But if you look at the T regulatory cell in tumor microenvironment, you see the glucose metabolizing enzymes expression goes down dramatically, and the fatty acid metabolizing enzymes is upregulated. So, what is the significance of this? So, we found that these cells, these T-regulatory cells, they express one molecule which is called CD36. This CD36 is nothing but a fatty acid scavenger receptor. So if you look at, if this T patient express low level of CD36 in these cells, this patient can survive more than 5 years. Nearly 100% of the patient survive. But if this patient express CD36, you see their survival probability goes down nearly 70%. And this very interestingly, these T regulatory cells are generated, and generation depends on the fatty acid metabolism. So if we block the glucose metabolism, you see the percentage of this T regulatory cell doesn't change. But if we block the fatty acid metabolic pathway by etoxomere, which is a fatty acid metabolism inhibitor, you see the number of T regulatory cell goes down. So if we block the fatty acid uptake by these cells doesn't grow. And very interestingly, if we grow the cells, T cell, in high fat diet, the number of cells, you see the percentage of this T regulatory cell goes up. If you let them grow in low fat diet, you see the percentage goes down. If you let them grow in low glucose diet, you see the percentage of these cells goes up, and in high glucose diet, this percentage of these cells goes down. So the fatty acid, if you change the fatty acid in the media, if you change from a high fat to low fat diet, the number of these T regulatory cells, you can reduce the number of these cells. So what is happening in the tumor microenvironment? If you have a limited glucose, and this tumor, they are highly proliferating, they used up most of the glucose, and very less glucose are left. And this effector T cell, they cannot get this glucose, so they cannot survive. So they die. So in tumor microenvironment, the effector T cell, they die. But these T regulatory cells, because they have the CD36, they express CD36, they use the fatty acid instead of glucose, because they don't want to compete with the tumor for the food, for the energy. They use a completely different metabolic pathway. They use fatty acid as their source of energy, and they survive. So, lipid metabolism and glucose metabolism, alteration of the glucose and fatty acid will decide whether this effector T cell will remain there, or they will convert into immunosuppressive T regulatory cells. So if you look at, this is a tumor, this is a mice, melanoma tumor containing mice. So this is the pattern of the volume of the tumor. If you give the mouse without any chemotherapy only low fat, high glucose diet, you see the tumor volume goes down. If you give them the high fat diet, you see that the pattern of the tumor goes up very rapidly and fast. If you apply chemotherapy along with the low fat diet, you see the tumor volume goes down dramatically. And if the mice were given high fat along with the chemotherapy, you see the chemotherapy doesn't work that much. So if you can change the pattern of the diet during the chemotherapy, you can get better option or better efficacy of the chemotherapy. Similarly, the work is going on in the our laboratory. Our preliminary data also showed that during the immunotherapy, if you change the food habit of the cancer patient, or if you give a high fat, high glucose, low fat diet during the immunotherapy, the T regulatory cell number goes down dramatically, and then in that case the immunotherapy as well as the chemotherapy work better to the cancer patient than the high fat diet patient. So in our second set, we have the information of the pattern of the non-coding RNA. So we try to analyze the non-coding RNA, and we identified a a series of non-coding RNA that are upregulated in the T regulatory cell. Very interestingly, by this volcano analysis, we could identify a number of microRNA that are upregulated or downregulated in T regulatory cells, and out of this, we could identify nearly 8 microRNA that are altered dramatically, significantly in T regulatory cells. We compared with different types of cells. In the T regulatory cells, this number of these microRNAs number or percentage goes down dramatically. And very interestingly, there is one microRNA which is called, I am not disclosing the number of this microRNA. I gave him the GSA3P. This microRNA goes down dramatically. And so we try to understand what is this microRNA. So we sequence this microRNA, and this is the structure of this microRNA. And very interestingly, this microRNA is present in all the primates, starting from rat to human to gorilla, chimpanzee, and they have a nearly 95% to 100% sequence homology. So we identified a microRNA that is dramatically reduced in T regulatory cell. So what is this microRNA is doing? We found that this microRNA, when this microRNA goes down, the T regulatory cells in tumor microenvironment goes up. And this is specifically downregulated only in T regulatory cells or not in other T cells. So, for that purpose, then we clone this microRNA and we develop two different vectors. One is the lentil virus vector, and another is the adeno-associated virus vector for a laboratory work, which is for the mice experiment. The lentil virus system was used for the laboratory experiment for the mice, and the adeno-associated virus was developed for the clinical trial. So, what is this microRNA is doing? So, we did lots of bioinformatics work and found that this microRNA targets a Treg specific transcription factor associated all the protein interotomies. So that these are the proteins which are required for the development and function of the T regulatory cell. So these are the target for this microRNA. So you can see this microRNA, if you overexpress this microRNA, the relative expression of this microRNA, the mRNA of different proteins goes down by the use of this microRNA. And if you use the anti-microRNA, the protein mRNA expression goes shoot up very dramatically. And very interestingly, this microRNA binds to the 3' UTR of all the proteins, all the Treg associated proteins. So we got a microRNA. If we overexpress this microRNA in the system, the Treg associated proteins which are required for the development of the T regulatory cell goes down dramatically. So we develop this microRNA in a lentil virus system and try to understand what is going on in the mouse model. So this is the microRNA. We develop a tetracycline regulated microRNA. So if you see this microRNA is if we apply no tetracycline, the microRNA is expressed highly. If we apply the tetracycline, there is a little bit of expression. So we applied this microRNA into a breast tumor model, mice model. You see, after 1 week, the tumor volume goes down without any chemotherapy. Only we injected this microRNA, the tumor volume goes down dramatically, nearly 60% in 1 week. And the T regulatory cell population in different immune compartment, like tumor size goes down, the T regulatory cells in the draining lymph node, peripheral circulation, tumor infiltrating lymphocytes, spleen and thymus, all the immune compartment goes down dramatically by the use of this microRNA. Then we used one melanoma model. You see, this is a mice, this is a tumor without virus, and if you apply this microRNA, the tumor volume goes down. Very interestingly, this is a multi-drug resistance adenocarcinoma tumor. You see, the tumor volume goes down dramatically with this microRNA. And very interestingly, there is, you can see the angiogenesis, which is developed because of the T regulatory cell develop secret lots of VEGF. So if we apply this microRNA, the T regulatory cell number goes down and you don't see the angiogenesis. And because of this non-availability of the food through the blood, the tumor die. So then we did the combinatorial therapy. One is that, you see, this is a mice tumor. If you don't apply any drug, that tumor goes grow very rapidly, and after certain time, all the mice they die. If you apply the chemotherapy, the tumor is restricted, but after certain time, the tumor goes shoot up very fast, and the mice die. If we apply this microRNA alone, you see the tumor volume goes. Most of the mice they die within 3 to 4 weeks of the tumor inoculation. But if you apply this microRNA, or if we apply this microRNA along with the chemotherapeutic drug, you see the tumor volume is so less. Even these mice survive more than 160 days. Our latest result is up to 175 days. These mice are still surviving. So that means these mice, after the tumor inoculation, they are surviving up to the 80% of their lifespan, because generally one mice they can survive from 190 to 200 days, but most of the mice they die after the tumor even in the presence of chemotherapy. Even if you apply the chemotherapy, or if you don't apply any chemotherapy, these mice die within 25 to 35 days. But if you apply this chemotherapy along with this microRNA, alone or in combination, these mice they are surviving. And if you look at the tumor volume, so you see this is the no drug, this is microRNA alone. And if you combine the chemotherapy and the microRNA, you see the volume of the tumor is dramatically reduced. And what is the status of the immune system? You see, this microRNA, it reduced the T regulatory cell population from here to here. It reduced the T regulatory cell. The type 2 cells, type 2 CD4, the from a type, because this tumor, during the tumor, the type 2 biasness is observed. So if you apply this microRNA, you see the type 2 cells number goes down. And type 1 cytotoxic CD8 cell, which are the cytotoxic, they are shooting up. That means it is reducing the type 2 cytokine producing T cells and inducing the CD8 cytotoxic T cell, IFN gamma producing T cells, so that this CD8 T cell then can attack the tumor. and they kill the tumor. And very interestingly, that type 2 macrophage, that is the M2 macrophage, percent IL-10 producing macrophage goes down dramatically because of this microRNA. That means this microRNA can rejuvenate our immune system very nicely, and they can handle our immune system so that our immune system now can work and attack the tumor. So I didn't show you the latest data from our laboratory. So in that case, when we applied the immunotherapy along with this microRNA, even we couldn't see some of this tumor in some mice. That means it is working better even in the presence of immunotherapy. So the immunotherapy works better if you can rejuvenate them by this microRNA. So this can be used as a potential adjuvant to the immunotherapy. So our aim is not to eradicate tumor, because we will not be able to cure cancer completely. But our aim is, can we increase the lifespan of a cancer patient? But similarly, like in mice, the most of the mice, they die if they develop cancer. They die within 4 to 5, 6 weeks. But when we apply this microRNA, they can live up to 175 days. That means 80%. If you convert into the human being, if someone develop cancer at the age of 25, if we apply this microRNA along with the chemotherapy, immunotherapy, or chemotherapy, this person can survive up to nearly 60 to 80 years. That means it's equivalent to a normal lifespan. So our intention is not to cure cancer. Can we make cancer from a fatal to a non-fatal disease? So the we after this chemotherapy or immunotherapy along with this microRNA therapy, someone will having cancer, they can say that I am surviving at the up to the age of 75 with my cancer. Thank you.
One kind of response like LPS can be used as therapy.
Yeah, people are trying to do that, but the problem with the LPS is, it's non-specifically activate all the cells. Not only Th1 cell, it can also activate the macrophage and others, and not it can activate the Th1 kinds of cell and also the Th2 cells. But in cancer patient, most of the cells became the T type 2 site type, Th2 type cells. If you activate the Th2 cells, that will be more detrimental uh, than not using the LPS. That's why the LPS is not a good option for use as a adjuvant to chemotherapy.
Thank you, sir. Now, I am requesting ma'am, our mentor and convener, Professor Inarai Banerjee, to give the ending note.
What an absorbing talk. Really, Gorida, salute. What an absorbing
Thank you.
talk. I enjoyed it so much. And especially for the students, every time you speak, it is like a storytelling. That's the most
Oh, thank you.
delightful part about your talks. So
Oh,
I
thank you.
will not keep you further because both of us will now have to go to
The
Suchanda and
other,
Tania. So I'm using our webinar platform to even acknowledge that. But we wish
yeah.
you all the best. Please continue with
Thank
your
you.
work many more years.
Thank you.
One last question, Gorida. What would be your advice to women? Because today is Raksha Bandhan. So for Raksha, prevention is better than cure. So,
Sure.
what would be your advice as an immunotherapeutic and cancer biologist?
As a cancer biologist, I would say keep your immune system boosting. Keep your immune system tight so that you can not only get rid of the cancer, our immune system can, protect you from the development of cancer, not only for the development of, even from the deadly infection like coronavirus. If you can keep your immune system boosted, your coronavirus cannot attack you. So better to boost your immune system from your natural diet component, not by using the artificial means. You boost your immune system by using the good microbiomes, by activating your microbiota, or by using some antioxidant containing food like fruits and natural vegetables that can protect your immune system. So go, try to avoid the junk food, use the natural food. So we call, I coined a term 25 years ago. Nowadays the people are use that term, is called nutraceuticals. I wrote a review in 25 years ago where we use the term nutraceuticals. Nowadays, we call it, people are using it to prevent different disease. From your nutrition, you can replace your pharmaceuticals from your nutrition. So I would like to use, particularly for the women, those are more prone to different types of cancer, like cervical cancer, and breast cancer. And most of my research work I did in breast cancer model, because the breast cancer is one of the deadliest cancer. And if we can tackle this cancer, we'll be able to cure. If breast cancer is a curable disease, but we cannot tackle it properly. uh, that's why I would say, to all the women, go for, if you feel any uncomfortable, go for mammogram to get a idea whether you are prone to, or you have started to developing tumor in your body or not. Tumor is not a deadly disease. When it became metastatic, that's after stage 3, it became metastatic. So if you can identify at the stage 1 or stage 2, all the breast cancers are curable, particular even the cervical cancer. So if you feel any discomfort in your breast or in your cervix, go to the doctor, check it up, and get rid of this deadliest disease.
On that positive note, we thank you, Professor Shah, for enriching our platform with your delightful research, and we wish you many more years of productive research, which will continue to save mankind. And to all
Thank you.
our
Thank you.
participants, thank you for joining us, and Shoheli for efficiently moderating tonight's show. With that, we shall end. Thank you
Thank
so
you.
much.
Thank you.
Thank you, ma'am. Thank you, sir.
Thank you.
Thank you,
Shall I
sir,
leave?
for.
Thank
Yes, sir. Yes, sir. You
you.
can. And to our audiences, you can post, you can obviously like our channel. You can share various videos with your friends and colleagues, and subscribe to our channel to go through all the videos which are archived in our channel. And you stay tuned for the next talk. And now, okay, thank you, and stay safe.